Akkam jirtu nagaa keessanii bakka jirtan maratti nageenyi keenya kabajaaf jaalalaa sinaa qaqqabu baga nagaan gara YouTube chaanalii kanatti dhuftan isiniin jecha YouTube chaanalii kanaaf haaraa warri taatan subscribe godhaatii maatii ta'a. Viidiyoo kana yoo jaallattan immoo like share gochuudhaan hiriyoota keessaniin akka qaqqabsiistan gamanumaan isin affeera. Har'aa baayoloojii kutaa 11ffaa Unit 3 Enzymes kan jedhu waliin ilaalla jechuudha. Unitii kana jalatti wantoonni nuti ilaallu yookaan immoo unit learning outcomes. These are the unit learning outcomes. You will define enzymes, or you will give the full definition of enzymes, describe the physical and chemical properties of enzymes, and factors affecting their activities. Waa'ee chemical physical properties of enzymes in ilaalla. Factors affecting their activities. There are several factors that affect the activities of enzymes, and also will explain mechanism of action of enzymes and their industrial application or application of enzymes in industry. Discuss enzyme kinetics. We will also discuss on enzyme kinetics. And finally, we will demonstrate proteins and their structures, because enzymes are basically proteins. Now, let's move into the next topic. What are enzymes? Or how do you define enzymes? Enzymes are protein molecules. They are made up of proteins, or they are made up of a chains of amino acids. And they act as biological catalysts. And we can also say biocatalysts. Enzyme only protein. They are protein molecules, and they are biocatalysts, or they are biological catalysts. What is the function of catalysts? Catalysts are something that accelerate the rate of chemical reactions by lowering activation energy. So enzymes are biological catalysts that speed up or accelerate the rate of a chemical reactions. But how do they accelerate the rate of chemical reaction? By lowering activation energy. We will talk about activation energy later in the next section. And all enzymes are proteins. All are proteins. They made of chains of amino acids linked together by peptide bonds. Enzymoonni hundi isaanii proteinidha. All enzymes are proteins, but not all proteins are enzymes. Enzymoonni hundumtuu proteinidha. Proteinoonni hundi garuu enzymii miti jechuudha. And there are 20 amino acids, 20 naturally occurring amino acids, and these amino acids are combined in different sequences to form different enzymes. Amino acidii 20tu jiru. Amino acidoonni 20 kun foormii sequeensii adda addaatiin combine ta'uudhaan enzymoota gosa adda addaa kennu jechuudha. So basically, all enzymes are proteins, and they are chains of amino acids linked together by peptide bonds. All cells contain different enzymes. Selling kamiyyuu enzymii qaba jechuudha. Depending on the type of the living cells and type of living cell and the type of organism. Based on this, but all cells and all organisms have their own enzymes which engage in tremendous biochemical activity called metabolism. These enzymes are take a part in metabolism. What is metabolism? Enzymoonni seeliin kamiyyuu enzymii adda addaa qaba. Enzymoonni kun biochemical activity garaagaraa keessatti qooda fudhatu jechuudha. So what is a metabolism? I hope you know this. Metabolism is the process of chemical and physical changes, or chemical and physical changes, or reaction, including the breakdown, catabolism, and synthesis or anabolism of molecules. So when we say metabolism, it is a general chemical reaction takes place in living cells. It may be breaking down of large molecules or synthesis of large molecules. If it is the breakdown of larger molecules, we call catabolism. Catabolism means breakdown. And if it is the formation of large molecules, we say this synthesis or anabolism. Anabolism. So metabolism is a combination of what? Catabolism and anabolism. So enzymes accelerate the reaction rates. They accelerate or they speed up the reaction rates, but they neither affect the nature of the products formed or nor undergo any changes by the reaction catalyzed. Enzymoonni ga'een isaanii chemical reactionii accelerate gochuudha yookaan immoo reetti chemical reactioniin itti gaggeeffamu ariifachiisuu yookaan si'eessuu yeroo ta'u, nature product sana maal hin godhan hin jijjiiran jechuudha. They neither affect the nature of products and also they undergo any changes by the reaction. They are not consumed by the reaction. They cannot change it by the reaction. Xumura reactionii sanarratti enzymiin sun hin jijjiiramu, productiin sunis hin jijjiiramu jechuudha. Fakkeenyaaf reactioniin tokko fakkeenyaaf enzymiin sucrase jedhamu sucroosii yookaan immoo daayisakaraayidii double sugarii kana gara monosakarayidiitti jijjiiruudha jechuudha. Enzymiin sun dhumarratti sanuma ta'a, productiin uumamus isuma sana ta'a jechuudha. Kanaaf the function of enzyme is accelerating the reaction rate or speeding up the reaction rate, but it doesn't change the nature of the products and also the enzymes not consumed during the reaction or they cannot be changed by the reaction. So the molecules upon which enzymes act are called substrates. Substrates are the chemical reactants. They are the chemical reactants upon which enzymes act. Wantoonni enzymiin irratti act godhan yookaan reactantiin sun maal jedhamu? Substrate jedhamu jechuudha. For example, from this picture, this is the enzyme molecule and this one is a substrate molecule. As you can see from this, the shape of the substrate molecule and the shape of the enzymes are complementary. They are complementary in shape. Shape is substrate kanaa fi enzyme kana complementary dha yookaan immoo complementary doesn't mean the same. Similar jechuu osoo hin taane complementary jechuudha. Fakkeenyaaf wantoota naannoo keenya jiran irraati yoon isinii ibse fakkeenyaaf moonyee bunaa fi muka isaa yoo fudhattanii ilaaltan sheeppii isaanii complementary dha jechuudha. Kanaaf sheeppiin substrateef enzyme complementary shape jedha. The same yookiin similar jechuu osoo hin taane wanta wal keessa galuu danda'udha jechuudha. And then when enzyme binds to the substrate molecule, enzyme substrate complex is formed. This is an intermediate. This is an intermediate product. Enzyme substrate complex. So enzyme substrating kun enzyme kanatti combine yookaan attach gaafa ta'u bakki kun active site jedhama. Active site is a specific region of the enzymes which combines with the substrate molecules. So enzymes have active site where the substrate molecule combines or binds. So substrates combine to the active site of this enzyme, and enzyme substrate complex and intermediate product is formed. And this combination causes stress on the bond of substrate molecules and in products are released from here. Enzyme product complex is formed and then products are leave the enzyme. Enzyme kana gadhiisanii ba'u productiin enzyme kun ammas substrate kana kan biroo fuudhee gara productiitti convert gochuuf ni danda'a itti fufa jechuudha. So enzymes are extremely efficient in speeding up reaction and they can catalyze up to several million reactions per second. yeroo gabaabduu keessatti yookaan immoo sekondii muraasa keessatti reactionii hedduu gaggeessuu ni danda'u jechuudha. So this is the definition of enzymes. So enzymes are, they are biomolecules or biological molecules which speed up a chemical reaction, and they do not change the products, and also they never change it by the reaction, or they remain unchanged at the end of the reaction. Dhuma reactionii sanarratti enzymoonni akkuma isaanii duraatti hafu jechuudha. And we have said enzymes are biological catalysts or biocatalysts. But basically, what are catalysts? What are catalysts? A catalyst is a substance that speeds up a reaction and remain unchanged. A catalyst is a substance that speed up or accelerating a reaction and remain unchanged at the end of the reaction. Catalystiin definition isaa kanadha jechuudha. By the way, catalystoonni living organismii keessa qofa osoo hin taane chemistry keessatti illee barataniitu jedhee yaada. They are substances which speed up a reaction and remain unchanged at the end of the reaction. Amaloota catalyst keessaa tokko kanadha jechuudha. They remain unchanged. They never changed by the reaction. And also they do not altered by the reaction. Ofii isaanis hin jijjiiraman, reactionii sanas hin jijjiiraman jechuudha. So in general, there is no overall change to the nature of the products, the energy change that takes place during the reaction. Nature product sana hin jijjiiran jechuudha. Akkasumas immoo energy fakkeenyaaf reactionii sun endothermic yookaan immoo exothermic reaction yoo ta'e energy sana hin jijjiiran. Endothermic isa ta'e exothermictiin geeddaran jechuudha. And also there is no overall change to the catalyst itself. Enzymiin sunis reactionii sanaan affect hin ta'u jechuudha. So not all biological catalysts are proteins. Remember, proteins are biological catalysts. I mean, enzymes are biological catalysts and enzymes are proteins, but not all biological catalysts are proteins. That means, in addition to proteins, there are other types of catalysts which are made from RNA molecules. So, recently, some RNA molecules can catalyze some biological reactions. Kanaafuu enzymoota irratti dabalataan molaakiilonni RNA tokko tokko reactionii catalyze gochuu danda'u. Isaan kana ribozymes jennee waamna jechuudha. Ribozymes, the RNA molecules which catalyze or speeding up some reaction, and we call them ribozymes. Now let's move into the physical properties of enzymes. Here are the physical properties of enzymes: denaturation, solubility, colloids, biocatalysts, enzyme activity, precipitation, molecular weight. These are the physical properties of enzymes, and let us explain all of these physical properties one by one. Denaturation. What does denaturation mean? Denaturation is the process of breaking the inter and intermolecular non-covalent bonds. The process or the breakdown of the inter and intermolecular non-covalent bonds that distort the shape and the active site of the enzymes. Basically, enzymes are made of a polypeptide chain. They are made of a polypeptide chains. And these polypeptide chains are connect or they bind together by the inter or intermolecular non-covalent bonds. And so the breakdown of that inter and intermolecular non-covalent bond due to some reason, such as high heat, alteration or change in the pH value, heavy metals, high salt concentrations, solvents, and other reagents. And due to these factors, when the intermolecular bonds break down and the shape of the enzyme is changed. And we call this denaturation. Enzymoonni protiniidha jenneerra. Polypeptide chainii lama sadii fi kanaa ol of keessaa qabaachuu danda'u jechuudha. Polypeptide chainii kun molecular gidduu isaanii boondiitu jira jechuudha. Boondii sun gidduu amino asidii peptide boondiitu jira. Akkasumas immoo boondonni kanneen biroo ionic boondiin faa jiru jechuudha. Hydrogen boondiin jira jechuudha. Boondonni kun gaafa isaan sababoota garaa garaatiin wal gagadhiisan maal jenna jechuudha? Denaturation jenna jechuudha. And denaturation causes the change of the shape of the active site of enzymes. Shaping active site sun ni jijjiirama, shaping enzyme sanas ni jijjiirama jechuudha. And this property of enzyme is known as denaturation. But what factors can cause denaturation? Enzymes are denatured by high heat or high temperature. For example, above 40 degrees Celsius. In human, the normal temperature or the optimum temperature in which enzymes work effectively is obvious, that is 37 degrees Celsius. If the temperature rises above this number, it causes denaturation of enzymes. Akka nama keessatti enzymoonni qaama keenya keessatti argaman normal body temperature yookaan 37 degrees Celsius keessatti hojjetu jechuudha. Effectively hojjetu. This is the optimum temperature for enzymes in human. So when temperature rises above this number, lakkoofsa kanaa olitti temperaturiin gaafa dabalu, high temperaturiin maal fida? Denaturation fida jechuudha. The second is alternation in the pH or change in the pH value. And also, is that too low or too high pH value causes the active site of enzymes to be changed, and also it slow down the function of enzymes. Hojii isaan enzymoon seeraan akkan hojjenne taasisaa. Kanaaf pH'n qaama keenya keessa jiru jijjiiramuun isaa jechuudha. Enzymoonni pH value adda addaa keessatti hojjetu. Some enzymes work effectively in acidic environment, others work effectively in basic, and others require neutral environment. So different enzymes have, they need different pH value. So any change in the pH value, or when the environment becomes too acidic or too basic, it causes denaturation. And also factors such as heavy metals, high salt concentration, solvents, and other reagents similarly cause denaturation of enzymes. So this is one of the physical properties of enzymes. The second is colloids, colloidal nature, colloids. The colloidal nature of enzyme is the tendency of having little or no dialysis across the semi-permeable membrane. Basically, the colloidal nature of enzyme is the result of the large size or high molecular weight. Enzymes are molecules with high molecular weight. Isa molecular weight guddaa waan qabaniif, or due to their large size, these enzyme molecules possess extremely low rates of diffusion and form colloidal system in water. And we call these colloids, or the colloidal nature of enzymes. The colloidal nature of enzyme means what? The tendency of having little or no dialysis across the semi-permeable membrane. They have low rates of diffusion, and they form colloidal system in water. Colloidal nature in enzymoonni molecular weight guddaa ni qabu. Sababni isaa large molecule, and they are made of a sequence of amino acids. So due to this, they cannot pass the semi-permeable membrane. Semi-permeable membrane cross gochuudhaaf low diffusion qabu jechuudha. Yookaan immoo salphaatti glukoosii, amino aciidota kanneen biroo yookaan immoo baayoomolekiilsii qaama keenya keessatti argaman kanneen biroo faana gaafa wal bira qabnu semi permeable membrane cross gochuudhaaf yookaan pass gochuudhaaf reetiin isaan qaban xiqqaadha jechuudha. That nature is known as colloids or colloidal nature of enzymes. So as I told you, the colloidal nature of enzyme comes from the large size or high molecular weight of enzymes. It is the result of large size or high molecular weight. Another property of enzyme is biocatalyst property. What is biocatalyst property? This refers or indicates very small quantities or small amount of enzyme is enough to convert large quantity of substrate. Very small quantity of enzyme is enough to convert large quantity of substrate and remain unchanged after the reaction. This is the biocatalyst property. The next is solubility. Solubility. Enzymes are soluble in water and also they are soluble in salt, diluted glycerol, and alcohol. This causes denaturation. That means when enzymes are soluble in these things and they lose their shape, when enzymes lose their shape, that is known as denaturation. Enzymoonni soluble gaafa ta'an nature isaanii gadhiisu yookaan immoo shape inactive site isaanii ni jijjiirama. This is another property of enzyme. The next is precipitation. Precipitation. Precipitation refers the separation of enzymes for analysis. Enzymes can be separated by using different aqueous or ethanol solvents. And enzymes are amphoteric. They can be precipitated by acidic and alkaline solution. Enzyme only amphoteric molecule. That means they can act as a base or an acid. Akka beezittis akka aciidittis act gochuu danda'u jechuudha. Because they are made of what? Amino acids. Amino acid irraa ijaaramanii structure amino acid gara dhumaatti ni ilaalla. Amino acid irraa waan ijaaramaniif akka acidiif akka beezitti yookaan amala acidummaas amala beezummaas ni qabu jechuudha. So we call them amphoteric molecules. So since they are amphoteric molecules, they can be precipitated by acid and alkaline solution, or they are capable of ionizing either as an acid or a base, depending upon the acidity of external solution. So what is enzyme precipitation? Enzymes precipitation refers the separation of enzymes for analysis. And they can be separated by aqueous or ethanol solvents. Or they can be precipitated by acid and alkaline solution. Alkaline solution. What makes enzyme or what enables enzymes to be precipitated by acid and alkaline solution? That is because of their nature. What is the nature of enzymes? Enzymes are amphoteric molecules. They act as an acid or a base because of the amino acids. Amino acid irraa waan ijaaramaniif amino acid immoo functional group lama qabachuudha. Amine group fi carboxyl group qaba jechuudha. Kanaaf akka beezittis akka acidittis act gochuu ni danda'u. Another property is molecular weight. Molecular weight. Enzymes are large biomolecules and they hold polypeptide chains or chains of polypeptide. And their molecular weights range from 12,000 to over 1 million. Imagine, molecular weight isaanii guddaadha jechuudha. Molecular weight guddaa qabu. And that means they have large size. So the enzyme molecules possess extremely low rates of diffusion across a semi-permeable membrane and for colloidal system in water. For example, bacterial ferrodexin, this is one of the enzyme. This is one of the smaller enzyme. which has a molecular weight of 6,000. Enzymoota jiran keessaa bacterial ferrodoxini smaller enzyme. Molecular weight isaa garuu it's about 6,000. And one of the largest enzyme is pyruvic dehydrogenase. This is another type of enzyme, largest enzyme, which has a molecular weight of this much. So enzymes have they have large molecular weight. Enzymatic activity. This property of enzyme refers the general catalytic properties of an enzyme. What is the general catalytic properties of enzyme? Speeding up chemical reaction and remain unchanged at the end of the reaction. But the enzymatic activity depends on several parameters. It depends on several factors such as temperature, pH, enzyme concentration, and substrate concentration. These are the factors which affect the activity of enzymes. They either increases the activities of enzyme or decreases the activities of enzymes. Enzyme temperature, pH, enzyme concentration, substrate concentration, activity isaanii yookiin hin dabala yookiin hin hir'ata jechuudha. For instance, if the temperature increases, temperature increase yeroo ta'u, the rate of chemical reaction or the activity of enzyme also increases to some extent. Temperature gaafa dabalu, enzymatic activity in dabala. Garuu hamma ta'e qofattidha jechuudha. Optimum temperature olitti yookaan hanga barbaadamu sanaa olitti temperature yoo dabale garuu maal fida jenne denaturation fida jechuudha. Kan dura isiniif ibsa jechuudha. What is denaturation? Denaturation in mind interii fi intermolecular bonding enzymoota sana connect godhee kan jiru sun addaan babba'u breakdown ta'uun denaturation jedhama jechuudha. So when denaturation happen, denaturation gaafa dhufu maaltu hin ta'u jechuudha. The change of the active site of enzyme is lost. So the enzyme cannot able to attach or binds to the substrate molecule. Shaping active site distort ta'e jechuun enzymiin sun substrate faana connect ta'uu hin danda'u jechuudha. Akkasumallee pH. Enzymes have their own optimum pH in which they work effectively. Bakka itti effectively iddootti hojjetan pH mataa isaanii qabu. For example, enzymiin maawuzii keessatti argamu salivary amylase pH mataa isaa danda'e qabachuudha. pH sun gaafa jijjiiramu shaping enzymes ni geeddarama. And also enzyme concentration and substrate concentration. So these parameters or these factors affect the catalytic properties or the enzymatic property. Enzymes show the highest activity at optimum temperature and optimum pH. They show the greatest activity, highest activity at optimum temperature and the pH. Temperature fi pH optimum ta'e yookaan mijataa ta'e keessatti activitiin enzyme high dha jechuudha. Garuu optimum temperature sanarraati yoo hir'ate yookiin yoo dabale denaturation fida. Don't forget this. And a low concentration of enzymes and the substrates slow down the enzymatic reaction. Concentration in enzyme substrate xiqqaadha yoo ta'e enzymatic reaction ni xiqqaata yookaan immoo in hir'ata jechuudha. So we have finished the physical properties of enzymes. The physical properties of enzymes. So what are the physical properties of enzyme? This one, they are around 7. Denaturation. Denaturation is the breaking down of the inter and the intramolecular bonds which connect the enzymes molecules together. And denaturation means when enzymes lost its shape or when the shape of active site is changed. Solubility. Solubility is it is another physical properties of enzymes. This indicates enzymes are soluble in water and other things. The molecular weight. Enzymes are larger biomolecules. They have greater or high molecular weight. And also enzyme precipitation is the other physical properties of enzymes, which refers enzyme precipitation or uh, enzyme precipitate gochuudhaan analysis adda addaadhaaf enzymoota bakka garaa garaatti adda baasu jechuudha. That means enzymes can be precipitated in acidic or alkaline solution. Enzyme activity is the overall enzymatic activity. It refers what enzymes can speeding up a chemical reaction by and remain unchanged at the end of the reaction. Another is biocatalysts. Enzymes are biological catalysts which speed up a chemical reaction and they are effective at catalyzing a reaction. Another physical property is colloids. Colloids. Colloids refers enzymes are due to their large size and high molecular weight. Enzymes have low rates of diffusion or they show no dialysis across the semi-permeable membrane. These are the physical properties of enzymes. Now let's move into the chemical properties. The chemical properties of enzymes. The chemical properties of enzymes includes sensitivity, regulations, specificity, catalysis, and reversibility. These are the chemical properties of enzymes. What is sensitivity? Heat and pH sensitivity. Enzymes are sensitive to heat or temperature and they are sensitive to pH. So sensitivity refers an enzymatic reaction to heat or temperature and the pH or acidity and basicity activated at optimum levels. By the way, enzymes are very sensitive to temperature and they are very sensitive to pH since they are protein molecules. They are sensitive to it. Enzymoonni hiitii fi pHdhaaf baay'ee sensitiiviidha yookaan salphaatti dinaacha ta'u jechuudha. Sababni isaa neechariin proteinii waan ta'aniif jechuudha. And the rate of an enzyme action increases with rise in temperature. The rate of an enzyme action or enzymatic action increases when temperature increases. But at higher temperature, the rate decreases rapidly. Enzymoonni reettiin isaanii yookaan immoo hojiin isaanii gaafa temperature'i dabalu in dabala jechuudha. But at higher temperature or extremely high temperature, the rate decreases. Reettiin isaanii extremely high yoo ta'e temperature'i reettiin isaanii decrease ta'a. Reettiin isaanii decrease ta'uu qofa osoo hin taane sheeppii isaaniillee dhabuu danda'u jechuudha. Enzymes are also pH sensitive and they are also sensitive to pH, which means highly acidic or highly basic nature or environment is not suitable for enzymes to survive and perform efficiently. Kana jechuun maal jechuudha seetan? Hiittii fi pH'n enzymoonni heat temperature'ii fi pH optimum ta'e yookaan immoo mijaa'aa kan ta'e barbaadu jechuudha. Fakkeenyaaf always the optimum the temperature or the the body temperature of human is 37 degrees Celsius. This is the optimum temperature for enzymes work in human. Nama keessatti temperature'iin kun 37 degrees Celsiusin optimum temperature'idha jechuudha enzymoonni akka isaan hojjetanii. Kanaaf temperature kanaa ol yoo deemu to the maximum above 40 yoo ta'e garuu kana keessatti maal ta'uu hin danda'an hojjechuu hin danda'an jechuudha. Temperature'iin xiqqaan when the temperature is low or a decrease in temperature decreases enzymatic activity. Temperature decrease yoo ta'e enzymatic activity decrease ta'a. Temperature increase yoo ta'e enzymatic activity increase that. But extremely high temperature causes a rapid decrease in enzymatic activity, and also it causes denaturation. Regulation. Enzymes, or the activity of enzymes, can be controlled in two ways. One, by using activator, something that increases the activity of enzymes, or inhibitor, molecules that inhibit or decrease the activity of enzymes. The next is catalysis. Catalysis is the process of the acceleration of a chemical reaction by a catalyst, speeding up a chemical reaction by a catalyst. So enzymes are biological catalysts. They are biocatalysts, and they possess high catalytic efficiency. And they can transform about 100 to 10,000 substrate per second. Secondly, tokko keessatti reaction amma kana gaggeessuu danda'u jechuudha. Yookaan immoo substrate amma kana transform gochuu danda'u. Garamitti? Gara productiitti jechuudha. The reactions catalyzed by the enzyme show about 103 up to 108 times faster the reaction rate in comparison to the non-catalyzed reaction. Listen, when you compare catalyzed reaction and non-catalyzed reaction, the catalyzed reaction by enzyme is faster than this much faster than the non-catalyzed reaction. By the way, reactioniin tokko tokko qaama keenya keessatti kan gaggeeffaman otuu enzaayimoonni jiran ta'ee waggaallee kan fudhatu ni jira jechuudha. Ji'allee kan lakkoofsisu jira jechuudha. Kanaaf qaama keenya keessatti wal nyaatiinsi garaagaraa jiru. Fakkeenyaaf nyaata hin nyaannaa, nyaanni sun hin bullaa'aa, gara niwutiraantii adda addaatti geeddarama. Innaa wal nyaatisoota adda addaa meetaaboliizimii qaama keenya keessa jiran kun otuu enzaayimiin hin jiru ta'eeti waggaattu kan hin geeddaramne jiraachuu danda'a. Ji'eeyyuu kan itti fudhatu jiraachuu danda'a jechuudha. Kanaaf si amma kana faastar chemical reaction tokko jechuudha. And the next is reversibility. What is reversibility? This is another chemical properties of enzyme, and it refers the ability of enzymatic biomolecules to catalyze various metabolic reactions, both forward and reverse directions. Some enzymes have ability to transform or to catalyze both forward and reverse reaction. They are capable of bringing about reversion in a chemical reaction. And the digestive enzymes catalyze the hydrolytic reaction, which are reversible. For instance, lipase. Lipase is one of the enzymes which converts fat into glycerol and fatty acid. Fatty yookaan immoo lipidii gara glycerolii fi fatty aciditti kan geeddaru. This is one of the digestive enzymes. And also this enzyme can also hydrolyze them into their component. Lipase, glycerolii, fatty acidii gara lipidiitti jijjiiruu danda'a yookaan gara fattiitti jijjiiruu danda'a. Akkasumas immoo fattii gara glycerolii fi fatty acidiitti jijjiiruu danda'a jechuudha. So this ability of enzyme is known as reversibility. Reversibility. Enzymes are capable of catalyzing both forward and reverse reaction. Reactionii kan gara lamaaniin iyyuu kan jiru jechuudha. Faattii uumuudhaaf akkasumas immoo faattii gara kompoonentii isaatti adda baasuudhaaf yookaan caccabsuudhaaf isa kanallee reverse kan ta'e wal nyaatinsa isa reverse ta'e catalyze gochuuf dandeettii ni qabu jechuudha. Specificity. Specificity describes how restrictive the enzyme is in its choice of substrate. By the way, enzymes are very specific in their action. They are so specific. Enzymes act on particular substrate. Particular enzymes or specific enzymes act on specific substrate only. And we say this specificity. Specificity. Specificity maal agarsiisa? Enzymoonni enzymiin tokko substrate tokko qofa faana connect kan inni yookaan enzymiin tokko reaction tokko qofaadha catalyze kan inni godhu jechuudha. So enzymes are specific to a particular type of reaction. They are specific, and this property of enzyme is known as specificity. And there are different types of specificity. The first is bond specificity. As you can understand from this name, this type of specificity indicates that enzymes are specific for a bond, and it is also said to be relative specificity. The enzyme will act on a particular type of chemical bond regardless of the rest of the molecular structure. For example, lipase. This enzyme lipase can hydrolyze ester bond between glycerol and fatty acid in fats. Enzyme lipase jedhamu kun bondii ester bondii irratti qofaadha act kan inni godhu jechuudha. Ester bondii glycerolii fi fatty acid gidduutti argamu jechuudha. Kanaaf specificity akka kanaa maal jenna? Bond specificity jenna jechuudha. Bond specificity is also called relative specificity. The other is group specificity. It is also called structural specificity of enzymes. Enzymoonni bond specificity qabu yookaan bondii tokko qofa irratti act kan godhan. And also some of them are group specificity or they exhibit group specificity. This means enzymes are specific for a group. What group? A specific functional group. They act on molecules that have specific functional group. Some are act on amino group and others are act on phosphate group or methyl group. For example, this enzyme pepsin. I hope you are familiar with this enzyme. This is one of the digestive enzyme which is found in the stomach. Pepsin hydrolyze the peptide bonds in with amino group and specifically to aromatic amino acids. So enzymes show group specificity. Group specificity is also known as structural specificity of enzymes. It indicates that enzymes are specific for a group, functional group or specific functional group. The next is substrate specificity. Enzymes are specific for a particular substrate. A particular enzymes acts on a particular substrate molecules. And they show a specificity toward one substrate and one reaction. This kind of specificity is high. And we can also say substrate specificity or absolute specificity. This type of specificity is high. Enzymoonni gosa substrate wajjin connect ta'an yookaan immoo reactionii tokko qofaaf illee specificidha jechuudha. For example, you know this enzyme trypsin. Trypsin hydrolyze peptide bonds involving carboxyl group of basic amino acid like arginine and lysine. Enzyme trypsin jennu kun maal hydrolyze godha yookaan immoo maal catalyze godha peptide bond irratti act godha jechuudha. Peptide bonds which involves carboxyl group of amino acids such as arginine and lysine. Kanaaf enzymiin kun kanarraa kan hafe kan biroo hydrolyze gochuu hin danda'u jechuudha. Peptide bondii irraa kan hafe kan bira bondii kan biraa irratti act gochuu hin danda'u. Kanaaf yeroo hundumaa substrate kana faanadha jechuudha connect ta'ee kan jiru. Yookaan immoo substratein kanneen biroo yoo jiraatan kan enzymiin kun irratti act godhu hin jiru yoo ta'e hin ta'a jechuudha hojii malee. Kanaaf amman taa'u jedhee enzyme kanneen biroo yookaan substrate kan biroo irratti act gochuu hin danda'u. That means enzymes are specific to substrate. They are specific to the substrate they act on. Another enzyme, chymotrypsin, hydrolyze peptide bonds of aromatic amino acid such as phenylalanine and tyrosine. Isaan kun inni kuniif kun waan isaan irratti waan isaan hydrolyze godhan yookaan catalyze godhan garaagarummaa qaba jechuudha. Isaan kun aminoo asiidii gartuu kan biraatti catalyze godha. Inni kun immoo aromatic aminoo asiidii warra jennu catalyze godha jechuudha. Optical specificity. This is when enzyme acts on the substrate optical configuration. Remember, enzymes are specific to the bond they catalyze, and also enzymes are specific to the substrate they act on. And enzymes are they are also specific to the substrate, the optical configuration of substrate. And this kind of specificity is known as stereospecificity. Stereo means the arrangement of atoms in the space. They are specific not only to the substrate but also to the optical configuration. For example, L-amino acid oxidase acts on L-amino acid, not on D-amino acids. L-amino acid, D-amino acid isomers isomers. Do you know isomers? They have the same molecular formula and but different in arrangement. You have learned this in chemistry, I hope. L-amino acid, D-amino acid isomerota jechuudha. Kana jechuun molecular formulaan isaanii tokko ta'ee arrangementiin yookaan akkaataa isaan itti arrange ta'an garaagarummaa kan qabu jechuudha. So enzymes are also specific to optical configuration of enzymes, optical configuration of the substrate. The next is cofactor specificity. Enzymes are not only specific to the substrate, but also they are specific to its cofactor. This is the greater specificity. In this, enzyme act specifically towards a specific substrate and cofactor. And the reaction is allowed if only the substrate and the cofactor are the same and match. But if the cofactor is absent, the enzyme remains inactive, even though many substrate molecules are present. Kun immoo enzymoonni cofactoriin isaanii yoo jiraate yookaan cofactoriin yoo hin jiru ta'e enzymiin sun inactive ta'a jechuudha. Kanaaf cofactoriin molecuulii enzymii irratti attach ta'uudhaan enzymii sun active akka inni ta'u taasisa. Enzymoonni tokko tokko cofactorii kan barbaadan ni jiru jechuudha. Kanaaf enzymoonni akka sana cofactorii maash ta'e isaan faana kan deemurraan kan hafe cofactorii kanneen biraatti connect ta'uu yookaan bind ta'uu hin danda'an jechuudha. This type of specificity is known as cofactor specificity. Cofactor specificity. So, so this is all about today's lesson. And today we defined enzymes. Enzymes are biological catalysts or they are biocatalysts that speed up a chemical reaction and remain unchanged at the end of the reaction. And catalysts are molecules that speed up a chemical reaction and remain unchanged. And enzymes or other catalysts do not change the product is formed, and also they do not consumed by the reaction. And also we have talked about the physical and the chemical properties of enzymes. In next topic, we'll talk about Unit 3 Part 2, the function of enzymes. Barnoonni keenya akkana fakkaata yeroo dhufu the functions of enzymes kan jedhu waliin ilaalla. Warri YouTube channel kanaaf haaraa taatan subscribe gochuudhaan maatii ta'a. Video kana yoo jaallattan like gochuudhaan hiriyoota keessaniif immoo share gochuun dagatina. Galatoomaa yeroo barnootaa gaarii.